Chapter 14, Glycolysis, Gluconeogenesis, and the pentose phosphate pathway. Section 1. Now we have to keep in mind that we're going to be looking at the macromolecules that we ingest in our daily diets. For whatever reason, we choose to focus our initial views of cellular respiration, the catabolism of what we take in, the nutrients, those macromolecules. We seem to focus on glucose first. And rightfully we should, because glucose is a central molecule that is going to be utilized in so many other aspects of cellular metabolism that aren't just energy production via pyruvate going to the mitochondria for citric acid cycle and oxidative phosphorylation. But we'll get to those later in this chapter and in other chapters. First, here in section 1, we're going to be looking at glycolysis, okay? Hopefully you remember from earlier courses that glycolysis is the breakdown of glucose, where glucose is degraded, broken down to form 2 3-carbon pyruvate molecules. In the process of this, a couple of ATP will be generated, and, well, I should say a few will be generated, and we'll talk more about those numbers in a minute. And then some NADs will be reduced to NADH. That's really what we need to focus on when we're talking energy here with glycolysis and cellular respiration. Initially, it's the NADH with that excited electron. This is where the real, true energy lies at. Now, glycolysis, we will rudimentarily split it into 2 parts, okay? There's the first part and a second part. The first part, or the first phase, we refer to as the preparatory phase. The preparatory phase takes glucose or any of the other sugars, we'll talk more about that later, takes this and immediately imbues some energy into the reaction mix. How does it do it? Well, ATP is hydrolyzed. ATP hydrolyzed down to a diphosphate, and that terminal phosphate is now added to glucose, hence glucose 6-phosphate. Glucose, though, is not the molecule that can easily be split in half, because remember, 1 gets split into 2, because however you go and try to slice this way, that way, that way, it doesn't matter because you can't end up with 2 halves that are approximately the same. They may be enantiomers, but it can't be done. So, going to take the energy that was imbued here, invested into this reaction, and going to use an enzyme, phosphohexose isomerase. Ends in A, so it's an enzyme. What prefaces it? Isomerase, isomer. It's an isomer making enzyme, sorry. What compound does it recognize? Phosphohexose, or if you're a biologist, glucose 6-phosphate. Okay, phosphohexose, glucose 6-phosphate, tomato. One's for the chemistry minded, the other's for the biology minded. Energy gets imbued into the system, this mix, via ATP here at step 1. Step 2, 6-sided ring gets turned into a 5-sided ring. Hey, look at this. I can split this down right there. Boom. As they're showing here. in the purple green. Are they perfectly matched? No, but they're pretty darn close. So invest some energy, take a 6-sided, turn it into a 5-sided, invest a little more energy. So now we have fructose, because remember it's 5-sided, not 6-sided anymore. Fructose, 1,6, carbon 1, carbon 6. Now both have phosphates. Bisphosphate. Fructose 1,6 bis by 2-phosphate. Where's the phosphates? Carbon 1, carbon 6. An aldolase is an enzyme that will cut this molecule in half. What you end up with? Glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. These two right here. And notice the structures are not set in stone. Okay? There seems to be some back and forth between G3P, which is acronym. The G3P is an acronym for G3P, glyceraldehyde 3-phosphate, and DAP, dihydroxyacetone phosphate, DAP. It goes and fluctuates between these two, its forms, based on hydroxyl to carbonyl, hydroxyl to carbonyl. So we get this back and forth, back and forth. This is the preparatory phase. Notice 1, 2 ATPs. 2 ATPs were invested into this. You're preparing for the split. The thing is, can't do anything with G3P, can't do anything with DAP. Need to take these and turn them into something else. Well, the second half, or the second phase, is known as the payoff. This is where you get something back. All right, what are we getting back? Well, turns out the DAP can't move forward, okay? The enzymes in the cytoplasm of the cell, whether it's prokaryote, eukaryote, doesn't matter, will utilize G3P as a substrate. So when DAP changes its structure, changes its shape to become G3P before snapping back, glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase removes hydrogen. We'll take any and all G3Ps and turn them into 1,3-bisphosphoglycerate. How does it do that? Well, easily. It takes a bunch of inorganic phosphates that are just floating around and just adds them. Boom. No real energy is imbued there. In the same time as it's doing this, it is taking that hydrogen right there, popping it off, and it is adding it to NAD. Thus, this gets oxidized, this gets reduced. NAD here gets reduced to NADH. Remember, hydrogens, as I've stated in many lectures before, hydrogens are nothing more than an excited electron with a proton coming along for the free ride. That hydrogen gets removed, so this incoming inorganic phosphate, PO4, it's probably going to be 3-, will do a nucleophilic attack on that carbon. This hydrogen will be released, so it's inorganic phosphate via oxygen, one of its oxygens, binding to carbon here. Next step, phosphoglycerate kinase takes 1,3-bisphosphoglycerates, pops off that just added phosphate group, adds it at ATP. This right here, this enzyme-based removing of a phosphate, adding it ATP, this is what is known as substrate level phosphorylation. Okay, this isn't high yield, lots and lots of numbers will be made like we're going to see in oxidative phosphorylation with the enzyme. This is, still, it's an enzyme, so it's still going to be fast, but it's just not as fast, just not as, efficient. Go through, and then as you notice, step 8, step 9, you're basically just rearranging the furniture. Still 1, 2, 3 carbons, 1, 2, 3. It's now who's got the phosphate group, goes from carbon 1 to carbon 2. Hydroxyl goes from carbon 2 to carbon 1, and things just keep getting moved around. And then here at the last step, phosphoenol pyruvate, PEP, we've seen that previous chapter. PEP gets that phosphate group, that inorganic phosphate stripped off, added to ATP. So technically, last slide, the preparatory or investment phase, depending on what textbooks you're using, utilizes 2 ATP, turns them to ADP. That gets everything, the ball rolling. This payoff, 2 NADs become 2 NADHs. 4 ADPs become 4 ATPs. And you end up with 2 pyruvates that will be utilized later. So the carbon skeleton, whether you're starting with glucose or fructose, we'll talk about that later, you're going to end with 2 3-carbon pyruvates. Those pyruvates still have a lot of energy in them. Okay, just because the phosphate groups were pulled off doesn't mean, oh, all done, all said and done, no more energy here. That phosphorylation, that substrate level phosphorylation, ADP to ATP, well, yeah, some of the energy is removed from this organic molecule added to ADP, ATP, in the making of ATP. But there's still a lot of energy left there. Right there, though, the NADH, that's going to be a big player in later reactions. So when you look at chemically what's happening, okay, we draw this out linearly. You see that it's 1, 2, 3, 4, 5, 6 carbons. Our terminal carbon here, carbon 6, with this glucose. This is where ATP is going to be hydrolyzed to add inorganic phosphate, giving us a diphosphate. In the process, glucose 6-phosphate needs to be turned into fructose. So how do we do this? Well, first off, need to take some of that energy we just got from the addition of the inorganic phosphate, and we need to move this carbonyl from carbon 1 to carbon 2. Okay. In step 3, this hydroxyl out here is going to be a great place for a nucleophilic attack from, again, a 3rd phosphate from ATP. So now we have a phosphate group on carbon 1 as well as carbon 6. We've also gone from glucose, the 6-sided ring of glucose, to the 5-sided ring of fructose. Now we need to cut. Step 4 is the cutting of fructose 1,6-bisphosphate. We're going to cut between carbons 3 and carbon 4. Again, remember I told you, are they exact copies of each other? No, they're more like mirror images of each other now. So they're enantiomers. These enantiomers, though, their shape is not set in stone, so there will be some back and forth conversion. Glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, back and forth, back and forth. Turns out, though, as I said, dihydroxyacetone phosphate, it's not a substrate for any enzyme that we're going to be paying attention to. Glyceraldehyde 3-phosphate is. So any G3Ps, native, or after DAP does its conversion to G3P, will immediately be snatched. It'll be snatched up. And then what you see here is that things are going to start getting stripped off. Okay? So you're going to have oxidative phosphorylation of glyceraldehyde phosphate. Going to have NAD reduced to NADH. Remember, strip off that electron / hydrogen. Also going to add on an inorganic phosphate. Now this isn't stripped off of ATP or anything. This is just a free floating in the cytoplasm. So now you end up with dual phosphates on carbon 1, carbon 3 of our glycerate molecule. Hydroxyl at carbon 2. Carbon 1 has a carbonyl and a phosphate. Carbon 3, phosphate group. Going to strip off this phosphate group, generate some ATP off of carbon 1. Boom. Go through. Now we need to do some rearranging. Phosphate group that was on carbon 3 is now going to be moved to carbon 2. Shift. Carbon 1, carbon 2, carbon 3. We're now going to get a double bond between carbon 2, carbon 3. And finally, we're going to strip off that third phosphate, making some more ATP. The overall equation we're going to see here. Glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphates. Remember, this is actually ATP. Yields 2 pyruvate, because remember, glucose gets split in half. 2 NADHs, because this NAD is going to be reduced. 2 free hydrogens. 2 ATP and 2 waters, because during the, some of the reactions of taking the G3P, turning it into pyruvate, you're going to release some different molecules here. And also of taking the inorganic phosphates and adding them to ADP, there's going to be some release there also. It's the reduction of NAD, okay, reducing it to NADH by the transfer of a hydride ion, okay. Hydride ion, that means it's a proton, 2 electrons. From the aldehyde group of glyceraldehyde 3-phosphate to the NAD, okay. This is where we're going to get more energy, maximal energy, in the mitochondria in later reactions. All of this is exergonic. Everything we talked about in the last slide. It's all, not every step along the way, but the sum total is exergonic. Going from glucose to pyruvate is exergonic because it's going to be, look at this, negative 146 kilojoules per mole. Quite a bit. You take the ATP, forming the ATP from A diphosphate and inorganic phosphate, which happens during the payoff phase. Oh, look at that, 61. Okay, that's very, very endorgonic. Have an extremely exergonic thing overall. Embedded within it, though, are going to be a couple of steps that are extremely endergonic. So I told you in chapter 13. You can go through and each of the steps calculate how much free energy is in this step, in this step, in this step. And then you add them all up, the pluses, the minus, delta G's. The sum total, negative 146 kilojoules per mole plus the 61 means you have a net negative 85 kilojoules. So even taking into account how much it costs to invest the energy into making of those ATPs, you still have a lot left. That's where you get the energy for the rearranging of the deck chairs. Changing from a 6-carbon ring to a 5-carbon ring is going to chip away at this. That energy stored in the pyruvate, there's still a lot there, okay? Pyruvate has not been exhausted of its energy. Aerobic processes, I keep referring to citric acid cycle and oxidative phosphorylation in the mitochondria. For some microbes, some eukaryote cells under certain conditions, us under certain conditions, are going to go anaerobic. We do anaerobic lactate metabolism. We'll talk about that more in a later section. This is a short-term thing for us. Many bacteria, this is their ultimate goal because they will not be using aerobic processes. Some things, like our friends, the yeast, like to make ethanol. Go beer. Now, if you think back, I keep saying those inorganic phosphates were added to imbue some energy into the system, but they're also there to keep those intermediates from leaving the system, leaving this series of reactions, maybe being exported out of the cell, moving out of the cell, because the phosphorylated versions of are highly polar. And if you think back to your membrane chapters from last semester or from any other course you've had where they discuss membranes, anything that's highly polar is highly soluble. Anything soluble that's highly soluble or polar cannot pass through membrane. So this keeps everything in the cell. So it can't leave the cell. It's by keeping them there, keeping them phosphorylated, keeps the energy, so we're not losing energy, because remember, you cutting off, adding those phosphates, there's a lot of energy there, boom, and if you just immediately removed it, well, second law of thermodynamics will apply very quickly. You're going to lose a lot, and instead of being 98.6 degrees Fahrenheit, 37 degrees Celsius, you're going to be a lot higher than that. But by keeping those phosphate groups there throughout multiple steps and then slowly picking away at stuff, here's some hydrogen, here's some more electrons, now we take off one of the phosphates, step, where, things get rearranged. Well, hey, guess what? We're going to be bleeding off some of the energy every time we take a carbonyl from carbon 1, move it to carbon 2, and move the phosphate group around. By the time you go and take off that last phosphate group at the end from PEP going to pyruvate, there's a lot less energy. Still a lot there, but there's it's less. So second law of thermodynamics isn't going to be quite the slap that it was. So the preparatory, which I keep inadvertently saying the introductory phase, or the investment phase. Different textbook, and that's the way I used to teach it for the freshmen. Got to use 2 molecules of ATP. So you can get this fructose 1,6-bisphosphate. Remember, fructose 1,6-bisphosphate, this is the one that's going to get split. Going to be cut between 3 and 4. So that way you yield 2 somewhat identical molecules. And you go through and you start looking at the different enzymatic reactions. Hexokinase is what activates glucose. It gives it some of the energy that's going to help power the next series of reactions. This hydroxyl group is going to be replaced by an inorganic phosphate that has been taken from ATP. Magnesium's there, remember, because we talked about in the last chapter, the magnesium interacts with the gamma, the beta, or the alpha, the beta, depending upon which ATP or ADP helps stabilize. So that way that those, the oxygens that are sticking off the inorganic phosphates aren't too interactive. So this helps stabilize everything. Incoming ATP or inorganic phosphate from ATP, you get a nucleophilic attack on this carbon here, carbon 6. It becomes an electrophile. You get the switching of, off it goes. So ATP is going to be our donor. This magnesium has to be there to help stabilize everything, okay? Low magnesium levels, this won't work. We like to draw this as being reversible, and we always think that most chemical reactions are reversible, but in biological settings in the cell, this is a one-way ticket. You can't just pop this inorganic phosphate off and just immediately add it back to ATP. No. Why? Well, it's negative 16 kilojoules going from this way. Going in the reverse means you're going to be this, but positive, and that's way too high to just inadvertently happen. Turns out we have lots of different hexokinases in us. Okay, we have 4 different ones. Hexokinase 1, 2, 3, and 4. They're numbered via Roman numerals. These are what are known as isozymes. Different genes, different enzymes that all do the same thing. For some reason, evolution seemed to decide we needed this. So those organisms that had multiple versions of this hexokinase, like enzyme, seem to do better, because for whatever reason, whatever cellular conditions in whatever cell and whatever tissue organ, that hexokinase 1 just doesn't seem to work. Guess what? You got 3 other versions that are also there ready to do the job. So if you're one of those early ancestral organisms that only had hexokinase 1 and you are a multicellular organism, and for some reason in your organs you were exposed to something that's going to inhibit or interfere with what we would now think of as hexokinase 1 activity, and you had no other versions, you're screwed. Those organisms that had 2 isozymes, one, they may have been exposed to the same thing, so hexokinase 1 was interfered with, but hey, hexokinase 2 is still there and can still do the job. They survived. Go through and you start looking at these different enzymes and what reactions they are, pushing through, I guess is the best way to talk about it. Phosphohexose, what we biologists would just call glucose-6-phosphate isomerase, or other people call it phosphoglucose isomerase. Okay, this is the taking of this 6-membered ring and turning it into a 5-membered ring. If you do the math, there's still 6 carbons, but carbon 1, carbon 2 are sticking up there like flags off of the ring. There is what they refer to as an enediol. I can't say it, tongue too thick. This intermediate. So there is an intermediate. You don't just immediately go and pop that one out and then boom, there you go. There's an intermediate that will be there. But you notice everybody still has the same number of hydrogens, still has the same number of hydroxyls. Nothing's been added, nothing's been removed. But the changing of this, 1.7 kilojoules per mole, okay, positive 1.7. So this movement was very endergonic. It's endergonic. Where'd the energy come to make so this can happen? Right there. That energy, along with this enzyme, pushed this change. So when you go through, and it's a little hard to see behind my big old head, so if you go and you look in the textbook, you'll see this better. Glucose-6-phosphate goes to, and you'll start to see things get switched. So the enzyme, here this is supposed to be, the active site within the enzyme. You have a nucleophilic attack from a R group for this hydrogen on C2, switching of the electrons here from C1 in this carbonyl group. Get a proton coming in. So now carbon 1 here, or I'm sorry, carbon 1 here now has a hydroxyl. Carbon 2, which had a hydroxyl and a hydrogen, now has just the hydroxyl. We now have a carbon-carbon double bond here between 1 and 2. Here's one of our intermediates. Carbon 1, which had a hydroxyl and a hydrogen, things get shifted around. That double bond, boom. Now you get that carbon here. Carbon 1 is doing a nucleophilic attack on one of the R groups within the active site. The same hydrogen that was just taken from carbon 2 is now under attack. Boom. Get the shifting of this bond between this hydrogen and this oxygen. It will be released, and you now have carbon 2 has a carbonyl group. Hydrogen's been released from the R group, and it's now here at carbon 1. So carbon 1 now has 1 hydroxyl, 2 hydrogens. Carbon 2 now has a carbonyl. Hey, look right here. Oxygen. So this carbonyl here, this double bond here will be shifted to carbon 5 right here. So you'll now have a bridge between carbon 2 and carbon 5. Look, 2, oxygen, bridge, 5. So it's now a 5-sided ring. Carbon 1, carbon 6 are now sticking up there. Anything in enzyme kinase means it takes inorganic phosphate, the terminal inorganic phosphate alpha ATP, adds it to its substrate. Well, only place really available that's open for a new phosphate to be added here is at carbon 1. Again, phosphofructose kinase 1, also known as the acronym PFK1, with magnesium to help stabilize everything. Again, nucleophilic, electrophilic attack, blah, blah, blah. Now you have phosphate groups at carbon 1 and carbon 6. And this whole thing generates what? Negative 14.2 kilojoules per mole. Extremely exergonic. That whole phosphofructose kinase 1, the addition of that second phosphate group. Okay, this is regulated. Okay, we don't want this just to happen whenever. Only happen under certain conditions when needed. What are those conditions? Well, one of them is when ATP is going down. When ATP supply is being depleted, it's going down, we need to push through glycolysis and oxidative phosphorylation to get some more ATP back. So it's not just the ATPs going away, it's the A diphosphate and A monophosphate levels are increasing in the cell. As we talked about in the last chapter, A diphosphate, A monophosphate have their own roles in the cell. They're not just a byproduct of the hydrolysis of ATP to get the high energy inorganic phosphate. So fructose 2,6-bisphosphate is our allosteric activator. If you have enough of this, ultimately this will go back up. If you're running out of this is low, this is being depleted, then you need to run through this step very quickly. Then there's that cleavage. We need to cut this in half. Aldolase, okay? Aldolases do the cutting of big organic molecules. Here it's cutting a ring. You're going to end up with 2 3-carbon things. Notice this, the delta G, positive 23.8. This takes a lot of energy. Good thing you have 2 energy molecules that have just been added into the system on the last enzymatic reaction. We call this a class 1 aldolase reaction, or class 1 aldolase reaction, because class 1s, this type of reaction is found in animals and plants. Class 2, which utilizes slightly different mechanisms. They don't use the shift base intermediate like we're going to be talking about showing here. This is fungi and bacterial. So what you see happening here, and I'm looking at my big screen here, which is why I'm not looking into the camera, pardon me. Fructose 1,6-bisphosphate enters into the aldolase active site, because remember this is an enzyme. It's an enzynase, so it's an enzyme. Multiple R groups from amino acids within are going to either do a nucleophilic attack or an electrophile, interaction. So this carbonyl is going to interact here, nucleophilic attack on the same carbon. So what was a carbonyl will form an intermediate of a hydroxyl, and then you get this nitrogen here from lysine has now formed an intermediate reaction, an intermediate bond with carbon 2 also. You also then get more interaction with the nitrogen back to lysine, and you start getting some bond switches here on carbon 3. Can't see it behind my big head, so I need to look in the textbook. So what we see happens is we have B here interacting, attacking on this hydrogen. You get this bond reforming, so you end up with a carbonyl here. And that's 1, 2, 3, 1, 2, 3. So this bond between carbon 3 and carbon 4, this electron pair we see shifts up between carbon 2, carbon 3. As we see, it's behind my head. 1/2, this half here becomes glyceraldehyde 3-phosphate. That we like. So that's carbon 3, carbon 4, 3, 4, 4, 5, 6. I was talking those wrong. So between 3 and 4 shifts up. So 4, 5, 6, there's G3P. But carbons 1, 2, 3 still aren't finished yet. We see the release from the different, the addition of water to break some of the bonds, and then you have the dihydroxyacetone phosphate released, and then you get resetting of the system during proton exchanges as hydrogens are shifted around. Dihydroxyacetone is not what our cells want, okay? Dihydroxyacetone phosphate, DAP, we want G3P. So you get this isomerase that's going to help push to G3P because this can't be used. This is substrate for the first set of enzyme reactions in the payoff phase, the second half. So the fate of the glucose carbons, carbons 1, 2, 3 will become the dihydroxyacetone, carbons 4, 5, 6 become G3P. G3Ps will then ultimately become carbons 1, 2, 3 for pyruvate at the end of glycolysis.